This is a presentation about 20 biotechnological advances prepared under the Transdisciplinary Doctoral Program in Science and Technology for Society in Sinvestov. Number 1. CRISPR-Cas systems and methods for altering expression of gene products. The invention provides for systems, methods, and compositions for altering expression of target gene sequences and related gene products. Provided are vectors and vector systems, some of which encode one or more components of a CRISPR complex, as well as methods for the design and use of such vectors. Also provided are methods of directing CRISPR complex formation in eukaryotic cells and methods for utilizing the CRISPR-Cas system. Number 2. CRISPR-based genome modification and regulation. The present invention provides methods for modifying chromosomal sequences. In particular, methods are provided for using RNA-guided endonucleases or modified RNA-guided endonucleases to modify targeted chromosomal sequences. Number 3. Methods of acquiring genome size and error. Provided is a method of acquiring genome size. The method comprises steps of sequencing random fragments of whole genome, acquiring all of k-mer information including k-mer depth, counting frequency of each k-mer depth value so as to determine expected k-mer depth, and acquiring the genome size by dividing the sum of k-mers by the expected k-mer depth. The method is convenient, rapid, and cost-effective. Also provided is a method of acquiring error of sequencing genome based on all of k-mer information including k-mer depth. Number 4. Filamentous pH encapsulating genome encoding ligand binding antibody comprising first and second polypeptides where first polypeptide is fused to carboxy terminal pH membrane anchor domain. Filamentous pH comprising the matrix of copy VIII proteins encapsulating a genome encoding first and second polypeptides of an antigenously assembling receptor, such as an antibody, and a receptor comprised of the first and second polypeptides surface integrated into the matrix via a filamentous pH coat protein membrane anchor domain fused to at least one of the polypeptides. Number 5. Method and system for generating a virtual progeny genome. Methods and systems for assessing the probabilities of the expression of one or more traits in progeny are described. Number 6. Methods for genome assembly and haplotype phasing. The disclosure provides methods to assemble genomes of eukaryotic or prokaryotic organisms. The disclosure further provides methods for haplotype phasing and metagenomics assemblies. Number 7. Nucleic acid construct comprising nucleic acid derived from genome of hepatitis C virus of genotype 1b, hepatitis C virus genome replicating cells transfected with the same, and method for producing infectious hepatitis C virus particles. A subgenomic replicon RNA, nucleic acid having an excellent autonomously replicating ability and a full genomic replicon RNA, nucleic acid having an excellent autonomously replicating ability and infectious HCV particle producing ability, each derived from a novel HCV of genotype 1b, are provided. Particularly, a subgenomic replicon RNA, nucleic acid, and a full genomic replicon RNA, nucleic acid, each derived from an HCV genome of the NC1 strain, which is a novel HCV genotype 1b isolated from a patient with acute severe hepatitis C, are provided. Number 8. Methods and means to modify a plant genome at a pre-selected site. Methods and means are provided to modify in a targeted manner the genome of a cotton plant using a double-stranded DNA break-inducing enzyme and embryogenic callus. Number 9. Genome shuffling method for autogamous plants utilizing dominant male sterility obtained by gene engineering technique and recurrent selection breeding system based on the genome shuffling method. A genome shuffling method for autogamous plants, including producing individuals having the following 3 traits and a tight coupling linkage by a gene engineering technique selected from a transgenic technique and a gene targeting technique, 1. Dominant male sterility, 2. Chemical tolerance, and 3. Lethality inducible by activating an inducible promoter, selecting, from progeny of the individuals, male sterile individuals by means of the chemical tolerance described in 2, and male fertile individuals by means of the lethality described in 3, arranging the male sterile individuals and the male fertile individuals close together in flowering periods thereof, so that the male sterile individuals are crossed with the male fertile individuals, harvesting seeds from the male sterile individuals, and repeating outcrossing using the seeds from generation to generation. Number 10. Intergenic regions as insertion sites in the genome of modified vaccinia virus Ankara, MVA. The present invention relates to novel insertion sites useful for the integration of exogenous sequences into the modified vaccinia Ankara, MVA virus genome. The present invention further provides plasmid vectors to insert exogenous DNA into the genome of MVA. Furthermore, the present invention provides recombinant MVA comprising an exogenous DNA sequence inserted into the new insertion site as medicine or vaccine. Number 11. Maize chlorotic dwarf virus genome and uses therefore. The present invention provides the nucleotide structure and organization of a novel maize chlorotic dwarf virus genome designated MCDVTN. Methods for using the complete or partial MCDVTN genomic sequence as a probe for diagnostic and other purposes are taught. Methods for inhibiting MCDVTN infection are also taught. These methods include the generation of transformed plants capable of expressing MCDVTN proteins, either in modified or unmodified form, and antisense sequences targeting MCDVTN genomic RNA. Recombinant production of MCDVTN proteins in appropriate host cells is also taught. Number 12. Full genome DNA of human cytomegalovirus strain JHC isolated from Korean patient and open reading frames thereof. Provided are a full genome DNA of a human cytomegalovirus HCMV strain JHC isolated from Korean patients and open reading frames ORFs thereof, and more particularly, UL1, UL119 and RL6. Number 13. Amplification and analysis of whole genome and whole transcriptome libraries generated by a DNA polymerization process. The present invention regards a variety of methods and compositions for whole genome amplification and whole transcriptome amplification. In a particular aspect of the present invention, there is a method of amplifying a genome comprising a library generation step followed by a library amplification step. In specific embodiments, the library generating step utilizes specific primer mixtures and a DNA polymerase, wherein the specific primer mixtures are designed to eliminate ability to self-hybridize and/or hybridize to other primers within a mixture, but efficiently and frequently prime nucleic acid templates. Number 14. Method for genome complexity reduction and polymorphism detection. The present invention provides methods to produce or reduce representation of a genome for sequencing and DNA polymorphism detection. In particular, the invention provides PCR-based methods, with normalization of the amplified products using a duplex-specific nuclease, in order to reduce overrepresentation of PCR products. Oligonucleotides for use in the disclosed method are also provided. Number 15. Biopesticide method for pest control. The present invention relates to a new method for pest control and/or for preventing or treating pest infestation. Said method comprises the inoculation of plants, parts of plants or the surrounding of said plants with an effective amount of endophytic Beauveria bassiana strains. In a further aspect, the present invention relates to the use of an isolated Beauveria bassiana strain having superior properties. Furthermore, biopesticides and compositions for pest control, in particular, for control of herbivorous insects and/or plant pathogens, are provided. Number 16. Methods and means to modify a plant genome. Methods and means are provided to modify in a targeted manner the genome of a plant in close proximity to an existing elite event using a double-stranded DNA break-inducing enzyme. Also provided are plants, in particular cotton plants showing tolerance to a field dose of at least 1 multiplication of at least 1 HPPD inhibitor, and methods for making such plants. Number 17. Social genome. Systems and methods are provided for processing a data stream in real time to identify connections between one or more social elements, and aggregating and storing the identified connections over a rolling time window. Social elements may be stored as nodes, and connection between social elements may be stored as labeled connections between nodes. Number 18. Using annotators in genome research. Provided are techniques for locating one or more genome patterns. One or more lexical annotators that each identify a sequence of nucleotides are created. One or more parsing rule annotators are created using at least one or more of the lexical annotators, 2, one or more dictionary entries, and 3, one or more previously defined parsing rule annotators. The one or more parsing rule annotators are used to discover the one or more genome patterns comprising a combination of the lexical annotators and the parsing rule annotators. Number 19. Methods for altering the genome of a monocot plant cell. Methods and compositions for altering the genome of a monocot plant cell and a monocot plant are disclosed. The methods and compositions use a double-strand break-inducing agent to alter a monocot plant or plant cell genomic sequence comprising a recognition sequence for the double-strand break-inducing agent. Number 20. High-throughput genome sequencing on DNA arrays. The present invention is directed to methods and compositions for acquiring nucleotide sequence information of target sequences using adapters interspersed in target polynucleotides. The sequence information can be new, e.g. sequencing unknown nucleic acids, resequencing, or genotyping. The invention preferably includes methods for inserting a plurality of adapters at spaced locations within the target polynucleotide or a fragment of a polynucleotide. Such adapters may serve as platforms for interrogating adjacent sequences using various sequencing chemistries, such as those that identify nucleotides by primer extension, probe ligation, and the like. Encompassed in the invention are methods and compositions for the insertion of known adapter sequences into target sequences, such that there is an interruption of contiguous target sequence with the adapters. By sequencing both upstream and downstream of the adapters, identification of entire target sequences may be accomplished. Thanks for your attention. Goodbye.